communication. Regulation of signaling pathways. Signaling pathways are crucial for cellular communication, allowing cells to respond to extracellular stimuli and coordinate appropriate intracellular responses. These pathways are tightly regulated to ensure proper cellular function and to prevent diseases such as cancer, autoimmune disorders, and metabolic conditions. The regulation of signaling pathways involves a variety of mechanisms, including the activation or inhibition of receptors. The regulation of signaling pathways involves a variety of mechanisms, including the activation or inhibition of receptors, enzymes, and second messengers, as well as feedback loops that maintain homeostasis. 1. Mechanisms of signal transduction regulation or receptor regulation. Desensitization and downregulation. Receptor activity can be regulated through desensitization, reduced receptor responsiveness to stimuli, and downregulation, receptor internalization and degradation. For instance, G-protein coupled receptors (GPCRs) undergo phosphorylation by G-protein receptor kinases (GRKs), which leads to the binding of beta-arrestins. This process uncouples the receptor from its G-protein, diminishing the signal transduction. Upregulation. In contrast, the number of receptors on the cell surface can be increased in response to prolonged low-level stimulation, e.g., hormone levels. This can enhance the cell's sensitivity to the signaling molecules. B-signal amplification and feedback. Positive feedback. In certain pathways, the output of a signal can amplify the initial signal. For example, in the MAP kinase pathway, activation of downstream kinases may activate upstream kinases, leading to further signaling enhancement. Negative feedback. Most signaling pathways are regulated by negative feedback mechanisms, which counteract or limit the signaling process. A common example is the PI3 kinase AKT pathway, where activation of AKT leads to phosphorylation and inhibition of upstream signaling proteins, limiting further activation. See crosstalk between signaling pathways. Cellular signaling is not linear but involves crosstalk between different pathways. Crosstalk allows integration of different signals in a coordinated cellular response. For example, signaling pathways involving CAMP, calcium ions, and phosphoinositides often intersect and modulate each other's activities, resulting in more precise regulation of cellular events such as cell division, metabolism, and gene expression. 2. Mechanisms of signal termination and degradation of signaling molecules. Second messengers, such as CAMP, CGMP, and IP3, are degraded by specific enzymes. For instance, phosphodiesterases PDEs degrade CAMP and CGMP to AMP, terminating their signaling activity. Receptor deactivation. Receptors undergo desensitization, reduced responsiveness, and internalization. For example, GPCRs can be phosphorylated by GRKs and then interact with beta-arrestins, which prevent further signal propagation and lead to receptor internalization. B-phosphatases and dephosphorylation. Many signaling pathways rely on phosphorylation and dephosphorylation events. Protein phosphatases remove phosphate groups from activated proteins, thus turning off the signaling pathway. For example, phosphatases like PP2A, protein phosphatase 2A, can dephosphorylate and inactivate components of the MEPK pathway, effectively terminating the signaling cascade. C. Receptor endocytosis and lysosomal degradation. Endocytosis of receptors, followed by their lysosomal degradation, is a common mechanism for terminating signal transduction. For example, epidermal growth factor receptor (EGFR) is internalized after ligand binding and then trafficked to lysosomes for degradation, thereby limiting the response. 3. Negative feedback loops and their importance. Negative feedback is crucial for maintaining cellular homeostasis. It acts as a mechanism to regulate and limit the duration and intensity of a signaling event. Several key examples include MEPK pathway. Negative feedback in this pathway occurs when the activated or extracellular signal regulated kinase phosphorylates and inactivates upstream components of the pathway, thereby reducing further signaling. PI3K AKT pathway. AKT activation can negatively regulate upstream signals like the PI3 kinase PI3K pathway by phosphorylation and inhibition of the receptor. Negative feedback ensures that once a signal has been received and an appropriate response initiated, the system does not remain in an active state for too long. This prevents overstimulation and protects the cell from Negative feedback ensures that once a signal has been received and an appropriate response initiated, the system does not remain in an active state for too long. This prevents overstimulation and protects the cell from potential damage. 4. Positive feedback and its role. While negative feedback is common, positive feedback can enhance or propagate a signal, ensuring that once a particular threshold is crossed, the response becomes irreversible. Examples include cell cycle regulation. Positive feedback loops are integral to the cell cycle. For instance, during the transition from G1 to S phase, the activation of cyclin-dependent kinases CDKs promotes further activation of CDKs, ensuring the cell progresses through the cycle. Immune response. Positive feedback is also observed in immune signaling, where cytokine production promotes further immune cell activation, enhancing the immune response. 5. Pathways regulated by hormones. Hormones play a significant role in regulating signaling pathways. Hormones such as insulin, glucagon, thyroid hormones, and steroids can modulate key signaling pathways, leading to a variety of cellular responses. Insulin signaling. Insulin activates the PI3K AKT pathway, promoting glucose uptake, glycogen synthesis, and lipid metabolism. Negative feedback occurs when increased glucose uptake leads to reduced insulin secretion. Steroid hormone signaling. Steroid hormones, such as cortisol or estrogen, regulate gene expression by binding to intracellular receptors that function as transcription factors. Feedback mechanisms control the synthesis of these hormones. 6. Regulation of gene expression through signaling pathways. Signal transduction pathways also regulate gene expression through activation of transcription factors. Examples include NF-kappa B pathway. NF-kappa B is activated in response to cytokine signals and leads to the transcription of pro-inflammatory genes. Feedback loops regulate its activation to avoid excessive inflammation. JAK-STAT pathway. In the presence of cytokines, JAK kinases activate STAT proteins, which translocate to the nucleus and promote gene expression. This pathway is regulated by negative feedback from inhibitory proteins such as SOCS, suppressors of cytokine signaling. Previous year questions with answers. Which of the following is a mechanism of terminating signaling in G-protein coupled receptor (GPCR) pathways? Activation of GRKs. B. Phosphorylation of G-protein C. Inhibition of phospholipase CD. Activation of beta-arrestins. Answer. Activation of GRKs. CSIR-Net Life Sciences, June 2017. The MEPK signaling pathway is regulated by which of the following mechanisms? A. Positive feedback by ERK. B. Negative feedback by MEC. C. Positive feedback by PKCD. Negative feedback by ERK. Answer. D. Negative feedback by ERK. CSIR-Net Life Sciences, December 2019. Which of the following is involved in the regulation of gene expression through signaling pathways? A. CAMP. B. CA2C. NF-kappa B. D. IP3. Answer. C. NF-kappa B. CSIR-Net Life Sciences, June 2020. This lecture provides a comprehensive understanding of the regulation of signaling pathways, ensuring you're prepared for the CSIR-Net Life Sciences exam. Let me know if you need further clarification or more topics. You said. Bacterial and plant two-component systems. ChatGPT said. Here is your high-quality, maximum detail CSIR-Net JRF Life Sciences lecture on bacterial and plant two-component systems. No tables, no wasted space. Fully elaborated from base to advanced. Includes previous year questions with answers in years. Structured for direct exam success. Bacterial and plant two-component systems. Two-component signal transduction systems (TCS) are essential mechanisms used by both bacteria and plants to sense and respond to environmental stimuli. These systems allow cells to adapt to changes in their surroundings, and they are vital for processes like stress responses, nutrient uptake, and virulence. The two-component system consists of a sensor kinase membrane-bound protein and a response regulator, usually a cytoplasmic protein, which interact to propagate the signal from the external environment to the interior of the cell. 1. Overview of two-component systems. The basic principle behind the two-component system involves the sensor kinase detecting an external signal and then transferring that information to a response regulator through phosphorylation. This phosphorylation event typically results in the activation or deactivation of the response regulator, which then affects gene expression, enzymatic activity, or other cellular processes. 2. Components of the two-component system of sensor kinase. The sensor kinase is typically a transmembrane protein that detects environmental changes. Upon encountering a specific signal, such as nutrient availability, temperature changes, or osmotic stress, the sensor kinase undergoes autophosphorylation at a histidine residue on its cytoplasmic domain. This phosphorylated sensor kinase then transfers the phosphate group to the response regulator. B response regulator. The sensor kinase is typically a transmembrane protein that detects environmental changes. Upon encountering a specific 1. Overview of two-component systems. The basic principle behind the two-component system involves the sensor kinase detecting an external signal and then transferring that information to a response regulator through phosphorylation. This phosphorylation event typically results in the activation or deactivation of the response regulator, which then affects gene expression, enzymatic activity, or other cellular processes. 2. Components of the two-component system of sensor kinase. The sensor kinase is typically a transmembrane protein that detects environmental changes. Upon encountering a specific signal, such as nutrient availability, temperature changes, or osmotic stress, the sensor kinase undergoes autophosphorylation at a histidine residue on its cytoplasmic domain. This phosphorylated sensor kinase then transfers the phosphate group to the response regulator. B response regulator. The response regulator is a cytoplasmic protein that receives the phosphate group from the sensor kinase. This phosphorylation induces conformational changes in the response regulator, enabling it to bind to DNA or interact with other cellular components. The action of the response regulator typically involves activation or repression of specific genes or modulation of enzymatic functions. Most response regulators contain a receiver domain responsible for receiving the phosphate group and a DNA binding domain responsible for regulating gene expression. 3. Mechanism of signal transduction is signal perception by sensor kinase. The signal is detected by the extracellular domain of the sensor kinase. In bacteria, this often involves interactions with environmental factors such as temperature, osmotic pressure, or the presence of chemicals like sugars or antibiotics. Once the signal is detected, the kinase undergoes conformational changes that allow it to autophosphorylate. B phosphorylation and signal transmission. The phosphorylated sensor kinase then transfers the phosphate group to a conserved histidine residue on the sensor's intracellular domain. This process is typically catalyzed by a histidine kinase domain. The transferred phosphate group is then passed to the response regulator, which typically contains an aspartate residue that is phosphorylated. C response regulator activation. Once the response regulator is phosphorylated, it undergoes conformational changes that allow it to activate or repress target genes, alter protein activity, or modify cellular processes. In some cases, the response regulator might dimerize, bind to specific DNA sequences, or interact with other cellular machinery to mediate the response. D signal termination. After the response regulator has executed its function, the signal is terminated by phosphatases that remove the phosphate group, returning the system to its basal state. In bacteria, this often involves dephosphorylation of the response regulator by phosphatase or by the sensor kinase itself, while in plants, phosphatases are also involved in terminating the response. 4. Bacterial two-component systems. In bacteria, two-component systems regulate a wide variety of processes, including virulence, stress responses, antibiotic resistance, and cell division. Bacterial TCS are often activated by environmental signals, such as nutrient availability, osmolarity, temperature, and the presence of toxic compounds. An example, the phoregulin. The phoregulin is a well-studied two-component system in Escherichia coli, involved in phosphate regulation. The system consists of the 4 sensor kinase, which detects low phosphate levels, and the PHO response regulator, which activates genes for phosphate uptake and utilization. In response to phosphate limitation, 4 autophosphorylates and transfers the phosphate to PHO, which then binds to DNA and activates gene expression. To phosphate limitation, 4 autophosphorylates and transfers the phosphate to PHO, which then binds to DNA and activates gene expression. The example, the NIVS-OMPR system. The NIVS-OMPR system in E. coli regulates the expression of outer membrane porins in response to osmotic stress. The NIVS kinase detects changes in osmolarity, and through phosphorylation, activates the OMPR response regulator, which then controls the transcription of OMPF and OMPC genes, which encode outer membrane proteins involved in solute transport. 5. Plant two-component systems. In plants, two-component systems are involved in regulating a variety of physiological processes, including growth, development, and stress responses. These systems are somewhat similar to bacterial TCS but are adapted for plant-specific needs. The AHK-ARR system in Arabidopsis. One of the best-studied plant TCS is the AHK-ARR system in Arabidopsis thaliana. The system includes the AHK Arabidopsis histidine kinase, sensor kinase, which detects cytokinin signals, and the ARR Arabidopsis response regulators. Cytokinins are plant hormones involved in cell division and differentiation, and the AHK-ARR system helps regulate responses to these hormones by activating or repressing gene expression in response to external signals. B. The ethylene signaling pathway. Ethylene is a plant hormone that regulates fruit ripening, leaf abscission, and stress responses. The ETR1 sensor kinase in Arabidopsis detects ethylene and regulates the activity of the ethylene-insensitive 2 response regulator. This regulation controls the expression of genes involved in ethylene responses, such as those involved in ripening and stress tolerance. 6. Role of two-component systems in pathogenesis. In both bacteria and plants, two-component systems play a critical role in responding to stress and environmental changes, making them integral to pathogenic processes. For instance, in plant pathogens, the two-component systems enable the pathogen to sense the host environment and modify its virulence accordingly. Similarly, in bacterial pathogens, TCS regulate the expression of virulence factors and antibiotic resistance mechanisms in response to host signals. Previous year questions with answers. Which of the following components is common to both bacterial and plant two-component systems? A. Histidine kinase. B. G protein C. Adenylate cyclase D. Calcium ion channels. Answer. A. Histidine kinase. CSI RNet Life Sciences December 2016. In the phoregulin of Escherichia coli, the sensor kinase 4 detects a change in phosphate levels and activates the response regulator PHO. This system is an example of A two-component signaling system. B. AMAPK cascade C. ACAMP signaling pathway D. A receptor tyrosine kinase pathway. Answer. A two-component signaling system. CSI RNet Life Sciences June 2018. Which plant hormone is primarily regulated by the two-component system involving AHK and ARR proteins in Arabidopsis? A. Cytokinin. B. Gibberellin C. Abscisic acid D. Auxin. Answer. A. Cytokinin. CSI RNet Life Sciences December 2019. This lecture provides a comprehensive understanding of bacterial and plant two-component systems, ensuring you're well prepared for the CSI RNet Life Sciences exam. Let me know if you need further clarification or more topics. You said. Light signaling in plants. ChatGPT said. Light signaling in plants. Light signaling in plants is a complex and essential process that regulates a variety of physiological responses, including germination, photomorphogenesis, flowering, and circadian rhythm. Plants use light as a signal to adapt to their environment, ensuring optimal growth. And development. Light signaling is primarily mediated through photoreceptors, which detect different wavelengths of light and translate these signals into molecular responses that alter gene expression and cellular processes. 1. Photoreceptors in plants. Photoreceptors are proteins in plants that sense light and initiate a signaling cascade to alter cellular behavior. These receptors are highly sensitive to specific light wavelengths, UV, visible light, and infrared, and they play a central role in photoreception and signal transduction. The main types of photoreceptors in plants include phytochromes. These receptors are sensitive to red and far red light and are involved in regulating processes such as seed germination, shade avoidance, and flowering. Cryptochromes, sensitive to blue and ultraviolet A UVA light. Cryptochromes regulate growth, circadian rhythms, and photoperiodic flowering. 1. Photoreceptors in plants. Photoreceptors are proteins in plants that sense light and initiate a signaling cascade to alter cellular behavior. These receptors are highly sensitive to specific light wavelengths, UV, visible light, and infrared, and they play a central role in photoreception and signal transduction. The main types of photoreceptors in plants include phytochromes. These receptors are sensitive to red and far red light and are involved in regulating processes such as seed germination, shade avoidance, and flowering. Cryptochromes, sensitive to blue and ultraviolet A UVA light. Cryptochromes regulate growth, circadian rhythms, and photoperiodic flowering. Phototropins. These are also blue light receptors that mediate phototropism, growth direction in response to light, and are important for stomatal opening and chloroplast movement. Safe loop, a protein that works with cryptochromes, also involved in the circadian clock regulation. 2. Phytochrome signaling. Phytochromes are a class of photoreceptors that absorb red and far red light. They exist in two interconvertible forms. PR, an active form, absorbs red light. PFR, active form, absorbs far red light. The interconversion between PR and PFR forms is key to how phytochromes regulate plant growth. When plants are exposed to red light, the PR form absorbs the light and converts to PFR. In the active PFR form, the phytochrome triggers downstream responses by interacting with specific transcription factors that regulate gene expression. Key roles of phytochrome signaling. Seed germination. Phytochromes play a crucial role in breaking seed dormancy, especially in response to light. For example, red light promotes the germination of many seeds, while far red light inhibits it. Shade avoidance. Phytochromes help plants detect shading by other plants. Under low light conditions, the phytochrome system promotes elongation growth, allowing the plant to escape from shaded areas. Flowering. Phytochromes are involved in the regulation of flowering in response to photoperiod, a process that determines when plants flower based on the length of the day and night. 3. Cryptochrome signaling. Cryptochromes are blue light receptors that mediate many aspects of plant growth and development. These photoreceptors are involved in processes such as photomorphogenesis, circadian rhythms, and flowering. Cryptochromes also interact with phytochromes to form a network of light-regulated signaling pathways. Key roles of cryptochrome signaling. Circadian rhythm regulation. Cryptochromes play an essential role in regulating the plant's internal clock, helping the plant anticipate changes in light conditions over a 24-hour cycle. Photomorphogenesis. Blue light detected by cryptochromes helps regulate growth patterns, including the elongation of stems and the opening of leaves, in response to light availability. Flowering time. Cryptochromes also influence the timing of flowering by interacting with other photoreceptors, particularly in response to light quality and duration. 4. Phototropism. Phototropism is the directional growth of plants in response to light. Plants tend to grow toward light, a process known as positive phototropism, or away from light, known as negative phototropism. This phenomenon is primarily regulated by phototropins, blue light receptors. Mechanism of phototropism. Phototropins sense the direction of light and initiate a signaling cascade that leads to differential growth on the shaded side of the plant. This unequal growth causes the plant to bend toward the light source. 5. Light and plant growth regulation. Light signaling in plants does not only affect the growth of individual cells but also coordinates the development of entire organs, such as leaves, stems, and flowers. The signal transduction mechanisms underlying light perception influence the expression of light-regulated genes that control cell division, elongation, and differentiation. Key processes regulated by light signaling. Leaf expansion and angle. Light influences the expansion and orientation of leaves to maximize light capture for photosynthesis. Stem elongation. Under low light conditions, phytochrome signaling activates genes that promote stem elongation, allowing the plant to reach for more light. Chloroplast movement. In response to light, chloroplasts move within plant cells to optimize light absorption or avoid excess light damage. 6. Light signaling and flowering. The regulation of flowering in many plants is influenced by the photoperiod, or the length of day and night, which is detected by photoreceptors. Phytochromes, in particular, are involved in mediating this process through a system known as the floral induction pathway. In short day plants, flowering is induced by long nights, while in long day plants, flowering occurs when the nights are shorter. Key pathways involved in flowering. FT flowering locus T. The gene FT encodes a protein that travels from the leaf to the shoot apical meristem, where it promotes the initiation of flowering. Phytochrome and cryptochrome signaling pathways interact with the FT gene in response to the photoperiod. 7. The role of light in stress responses. In addition to developmental processes, light signaling plays a role in how plants respond to stress. Photoreceptors can modulate the plant's response to abiotic stresses like drought, salt, and temperature. In these cases, light signaling helps adjust the plant's growth to conserve energy and resources. Previous year questions with answers. Which of the following photoreceptors in plants is responsible for regulating circadian rhythms and growth in response to blue light? A. Phytochrome. B. Cryptochrome. C. Phototropin. D. Safe loop. Answer. B. Cryptochrome. CSIR Net Life Sciences June 2017. Phytochromes in plants are sensitive to which types of light? A. Blue and ultraviolet light. B. Red and far red light. C. Green and yellow light. D. Infrared and X-rays. Answer. B. Red and far red light. CSIR Net Life Sciences December 2018. In Arabidopsis thaliana, the photoreceptor that mediates phototropism, directional growth in response to light, is a phytochrome. B. Cryptochrome. C. Phototropin. D. Safe loop. Answer. C. Phototropin. CSIR Net Life Sciences June 2019. This lecture on light signaling in plants covers the essential components and mechanisms, ensuring you are well prepared for your exam. Feel free to ask for more topics or clarifications. You said. Bacterial chemotaxis. ChatGPT said. Bacterial chemotaxis. Bacterial chemotaxis is a process by which bacteria move toward or away from chemical stimuli in their environment, a phenomenon that is essential for bacterial survival, colonization, and pathogenesis. It is a well-orchestrated cellular behavior that enables bacteria to respond to gradients of attractants, e.g., nutrients, or repellents, e.g., toxins. Chemotaxis plays a pivotal role in bacterial motility, helping them to navigate through complex environments and reach favorable conditions for growth or away from harmful substances. 1. Overview of bacterial chemotaxis. Chemotaxis in bacteria involves a series of steps that enable the bacterium to detect chemical gradients, process a signal, and then change its direction of movement. The process is mediated by a combination of sensory receptors, signaling pathways, and motor proteins that orchestrate bacterial movement. Bacteria typically employ flagella, which are protein structures that function as molecular motors to propel the bacteria toward or away from specific stimuli. 2. The chemotaxis mechanism. Sensing chemical gradients, bacteria possess specific chemoreceptors that detect changes in the concentration of chemicals in the environment. These receptors are usually located in the cytoplasmic membrane and are sensitive to attractants such as sugars, amino acids, and repellents such as toxins, harmful chemicals. The chemoreceptors can detect gradients, allowing the bacteria to sense whether the concentration of the substance is increasing or decreasing in their environment. Signal transduction pathway. When a chemoreceptor detects a change in the concentration of an attractant or repellent, it activates a signal transduction pathway inside the cell. The pathway typically involves a set of proteins, including Chi, Chu, Che, and Che, which collectively modulate the bacterial movement by controlling the rotation of the flagella. Chi is a histidine kinase that becomes autophosphorylated in response to a signal from the chemoreceptor. Che is a response regulator that, when phosphorylated by Chi, interacts with the flagellar motor to alter the direction of bacterial movement. Che dephosphorylates Che, reversing the movement. Flagellar motor and movement. The movement of the flagella is governed by a proton gradient, which provides the energy for flagellar rotation. Flagellar motor and movement. The movement of the flagella is governed by a proton gradient, which provides the energy for flagellar rotation. When the concentration of attractants increases, the bacterium alters its flagellar rotation to initiate running, a straight line motion. Conversely, when repellents are detected or the concentration of attractants decreases, the bacterium switches to tumbling, which involves random changes in the direction of movement, helping the bacterium to reorient and change its path. 3. Types of bacterial movement. Flagellar motor and movement. The movement of the flagella is governed by a proton gradient, which provides the energy for flagellar rotation. When the concentration of attractants increases, the bacterium alters its flagellar rotation to initiate running, a straight line motion. Conversely, when repellents are detected or the concentration of attractants decreases, the bacterium switches to tumbling, which involves random changes in the direction of movement, helping the bacterium to reorient and change its path. 3. Types of bacterial movement. Run. During this phase, the flagella rotate in a coordinated manner, pushing the bacterium in a straight line. This movement is associated with a clockwise CW rotation of the flagellar motor. Tumble. In response to a change in the concentration of attractants or repellents, the bacterium stops running and tumbles by reversing the direction of flagellar rotation. This random change in orientation allows the bacterium to reorient and assess the chemical environment again. This cycle of running and tumbling enables the bacterium to move toward areas with higher concentrations of attractants and away from repellents. The overall movement of the bacterium, therefore, appears as a biased random walk, where the bacteria tend to move toward favorable conditions over time. 4. Chemotactic signaling pathway. The chemotactic signaling pathway involves several key components. Methyl-accepting chemotaxis proteins, MCPs. These are the receptors that detect the presence of attractants or repellents in the environment. MCPs bind to specific chemicals, triggering the chemotactic signal transduction pathway. Qi and Chu. These proteins form a complex with the MCPs and mediate the phosphorylation of Che in response to changes in the concentration of chemicals. Che. Upon phosphorylation, Che interacts with the flagellar motor and induces a tumble, random rotation, when bound to the motor. Che dephosphorylates Che, leading to the cessation of tumbling and enabling the bacterium to resume running in a new direction. Adaptation. The bacterial system must also adapt to sustain signals. If a bacterium remains in the presence of a particular Qi and Chu. These proteins form a complex with the MCPs and mediate the phosphorylation of Che in response to changes in the concentration of chemicals. Che. Upon phosphorylation, Che interacts with the flagellar motor and induces a tumble, random rotation, when bound to the motor. Che dephosphorylates Che, leading to the cessation of tumbling and enabling the bacterium to resume running in a new direction. Adaptation. The bacterial system must also adapt to sustain signals. If a bacterium remains in the presence of a particular attractant or repellent, the sensitivity of the receptors is adjusted through a process called methylation, additions of methyl groups to the receptors allowing the bacterium to reset its response. 5. Types of stimuli in chemotaxis. Attractants. These are chemicals that bacteria move toward. Common attractants include nutrients such as glucose, amino acids, and oxygen. Bacteria often sense the presence of these attractants through an increase in their concentration in the environment. Repellents. These are chemicals that bacteria move away from. Toxic substances such as acids, heavy metals, and certain antibiotics often act as repellents. By moving away from these harmful substances, bacteria can avoid damage. 6. Significance of chemotaxis in bacterial behavior. Chemotaxis is critical for bacterial survival and pathogenicity. It allows bacteria to find nutrients. Chemotaxis enables bacteria to locate nutrient-rich environments for growth. Avoid harmful environments. By moving away from toxic substances, bacteria can evade damage and enhance their survival. Colonize hosts. In pathogenic bacteria, chemotaxis helps direct them to specific tissues in the host, aiding in infection and disease progression. Form biofilms. Some bacteria use chemotaxis to aggregate in specific locations, forming biofilms, which offer protection and help them persist in hostile environments. Previous year questions with answers. Which of the following proteins is directly involved in the regulation of bacterial flagellar rotation during chemotaxis? A. Chi. B. Chu. C. Che. Deep methyl-accepting chemotaxis protein. MCP. Answer. C. Che. CSIR Net Life Sciences December 2016. The bacterial flagellar motor rotates clockwise to initiate which of the following behaviors? A. Running. B. Tumbling. C. Mating. D. DNA replication. Answer. B. Tumbling. CSIR Net Life Sciences June 2017. Which component of the bacterial chemotaxis pathway is responsible for detecting changes in the concentration of attractants and repellents? A. Che. B. Chi. C. Methyl-accepting chemotaxis proteins. MCPs. D. Flagellar motor. Answer. C. Methyl-accepting chemotaxis proteins. MCPs. CSIR Net Life Sciences December 2018. This lecture on bacterial chemotaxis covers the fundamental concepts and mechanisms, ensuring you are well prepared for related questions in your exam. If you need further clarification on any point, feel free to ask. You said, quorum sensing. ChatGPT said, quorum sensing. Quorum sensing is a bacterial communication process that allows bacteria to coordinate their behavior in response to changes in their population density. It involves the production, release, and detection of signaling molecules called autoinducers. When the concentration of these signaling molecules reaches a threshold level, bacteria can synchronize their gene expression and behavior to perform tasks that are more effective at higher cell densities, such as biofilm formation, virulence factor production, and antibiotic resistance. 1. Mechanism of quorum sensing. Quorum sensing operates through the following basic steps. Production of autoinducers. Bacteria synthesize and release small signaling molecules called autoinducers into their environment. In gram-negative bacteria, the most commonly studied autoinducers are acylhomoserine lactones (AHLs), while gram-positive bacteria often use peptide-based signals. Some bacteria also use other molecules like In gram-negative bacteria, the most commonly studied autoinducers are acylhomoserine lactones (AHLs), while gram-positive bacteria often use peptide-based signals. Some bacteria also use other molecules like AUTOINDUCER2, AI2, a universal signaling molecule produced by both gram-negative and gram-positive bacteria. Detection of autoinducers. Bacteria have specific receptors that can detect the presence of autoinducers. These receptors are usually membrane-bound proteins or cytoplasmic proteins that bind to the autoinducers. In gram-negative bacteria, the receptor for AHLs is typically a protein called Luxor or its homologs. In gram-positive bacteria, peptide-based autoinducers are detected by the two-component signal transduction systems. Signal transduction and gene regulation. When the concentration of autoinducers in the environment reaches a certain threshold, indicating a high bacterial population density, the signaling molecules bind to the receptors. This binding activates a cascade of intracellular events that result in the regulation of specific genes. These genes may be involved in processes like biofilm formation, virulence factor production, antibiotic resistance, and other behaviors beneficial to the bacterial community. Coordinated behavior. Once quorum sensing reaches a threshold concentration, the coordinated expression of genes leads to behaviors that are advantageous only at high cell densities, such as virulence factor expression in pathogens or bioluminescence in certain marine bacteria. 2. Types of quorum sensing systems. Gram-negative bacteria. In these bacteria, quorum sensing is usually mediated by acylhomoserine lactones (AHLs). The AHLs bind to a receptor protein like Luxor, which then activates the transcription of target genes. A well-known example is the Luxor system in Vibrio fischeri, which regulates bioluminescence. Luxi produces the all-signal autoinducer, and Luxor binds to the signal to initiate gene expression once the concentration of all reaches a threshold. Gram-positive bacteria. These bacteria typically use peptide-based signaling molecules. One example is the CompuConnect system in Streptococcus pneumoniae, where the production of peptides autoinducers is regulated through a two-component system. In response to the signaling molecules, a regulatory protein activates the expression of genes related to competence and virulence. AI2-based quorum sensing. Some bacteria, including both gram-positive and gram-negative species, use AI2 as a universal signaling molecule. This molecule is synthesized from S-adenosylmethionine (SAM) and is detected by a receptor called LSRB in Escherichia coli. AI2 is involved in regulating a variety of behaviors, including biofilm formation and virulence in multiple species. 3. Functions of quorum sensing. Biofilm formation. One of the most important functions of quorum sensing is biofilm formation. Biofilms are clusters of bacteria embedded in a self-produced extracellular matrix, which provides protection against environmental stress, host immune responses, and antibiotics. Quorum sensing coordinates the expression of genes involved in the production of extracellular matrix components that form the biofilm. Virulence factor production. Quorum sensing regulates the production of virulence factors in pathogenic bacteria. For example, in Pseudomonas aeruginosa, quorum sensing regulates the production of toxins, enzymes, and other factors necessary for infection. When the bacteria reach a critical population density, they activate the expression of genes that help in host colonization and immune evasion. Antibiotic resistance. Quorum sensing also plays a role in antibiotic resistance. In some bacteria, quorum sensing regulates the production of enzymes that degrade antibiotics or the formation of biofilms that protect bacteria from the action of antibiotics. Bioluminescence. In some marine bacteria, such as Vibrio fischeri, quorum sensing regulates bioluminescence, allowing bacteria to emit light when they are in a dense community, which is used for communication with other organisms or for predation. Swarming and motility. In certain bacteria, quorum sensing can also regulate the movement of bacterial cells, leading to swarming or the coordinated movement of cells on surfaces. This behavior is critical for bacterial colonization. 4. Clinical implications of quorum sensing. Pathogenicity. Quorum sensing is crucial for the pathogenicity of various bacteria, including Staphylococcus aureus, Pseudomonas aeruginosa, and Vibrio cholerae. In these bacteria, the expression of virulence factors is tightly regulated by quorum sensing, and disrupting this system can reduce pathogenicity. This has made quorum sensing an attractive target for novel antimicrobial therapies. Antibiotic development. Inhibiting quorum sensing pathways, known as quorum quenching, has become a potential strategy to combat bacterial infections. By blocking the signaling molecules or their receptors, the expression of virulence factors can be suppressed without directly killing the bacteria, which reduces the risk of developing resistance. Previous year questions with answers. Which of the following is responsible for quorum sensing in Vibrio fischeri? A. AI2B acylhomoserine lactones AHLs. C. Peptides. D. Luxor. Answer. B. Acylhomoserine lactones AHLs. CSIR Net Life Sciences December 2016. In gram-positive bacteria, quorum sensing usually involves A. Acylhomoserine lactones B. Peptide-based autoinducers. C. AI2 signaling molecules D. Luxor proteins. Answer. B. Peptide-based autoinducers. CSIR Net Life Sciences June 2018. What is the main function of quorum sensing in pathogenic bacteria? A. Regulation of cell division B. Biofilm formation and virulence factor production C. Antibiotic resistance. D. Energy production. Answer. B. Biofilm formation and virulence factor production. CSIR Net Life Sciences December 2017. This lecture on quorum sensing provides a comprehensive overview of its mechanisms, functions, and significance in bacterial behavior, ensuring you are well prepared for related exam questions. If you need further clarification, feel free to ask. Search. Cellular communication and regulation of hematopoiesis. Hematopoiesis is the process of blood cell formation, which occurs primarily in the bone marrow in adults, and is regulated by intricate cellular communication mechanisms. The regulation of hematopoiesis involves several signaling pathways, growth factors, cytokines, transcription factors, and interactions between hematopoietic stem cells (HSCs) and their microenvironment. Cellular communication and regulation of hematopoiesis. Cellular communication and regulation of hematopoiesis. Hematopoiesis is the process of blood cell formation, which occurs primarily in the bone marrow in adults, and is regulated by intricate cellular communication mechanisms. The regulation of hematopoiesis involves several signaling pathways, growth factors, cytokines, transcription factors, and interactions between hematopoietic stem cells (HSCs) and their microenvironment. Cellular communication in hematopoiesis is a complex system where both intrinsic and extrinsic factors orchestrate the differentiation, proliferation, and survival of various blood cells, including red blood cells (RBCs), white blood cells (WBCs), and platelets. At the core of hematopoiesis are hematopoietic stem cells (HSCs), which are multipotent cells capable of self-renewal and differentiation into all blood cell types. The regulation of HSCs is crucial for maintaining hematopoiesis, and this regulation occurs through a combination of signals from the bone marrow niche and from circulating cytokines and growth factors. These signals act by binding to specific receptors on the surface of the hematopoietic cells, leading to various intracellular responses that regulate the cell cycle, differentiation, and survival of hematopoietic progenitors. Key players in hematopoiesis regulation. Cytokines and growth factors. These soluble molecules play a critical role in hematopoiesis by regulating the proliferation, differentiation, and survival of hematopoietic progenitor cells. The major growth factors include stem cell factor SCF, a key regulator of HSC survival and self-renewal. SCF binds to the c-KIT receptor CD117 on HSCs, promoting stem cell maintenance. Interleukins IL-3, IL-6, IL-11. These cytokines regulate the survival, proliferation, and differentiation of various progenitor cells. IL-3, for instance, promotes the proliferation of early progenitor cells, while IL-6 and IL-11 are involved in the differentiation of more committed progenitors. Granulocyte colony stimulating factor GCSF. This cytokine promotes the production of neutrophils and stimulates the mobilization of hematopoietic progenitors from the bone marrow into peripheral blood. Erythropoietin EPO, a glycoprotein that stimulates the production of erythrocytes, red blood cells, in response to low oxygen levels. Thrombopoietin TPO. This factor regulates platelet production by stimulating megakaryocyte differentiation. Transcription factors. These are crucial for controlling the differentiation of hematopoietic progenitors into specific blood cell types. Some important transcription factors include 2.1. A transcription factor that plays a significant role in the differentiation of myeloid and lymphoid progenitors. It is critical for the development of macrophages, granulocytes, and dendritic cells. GATA1. Essential for erythroid differentiation. GATA1 regulates the expression of genes required for red blood cell production. CEBP alpha and CEBP beta. These factors are involved in the differentiation of granulocytes and macrophages. Notch signaling. Notch signaling is a key pathway that regulates lineage commitment of HSCs. It can either promote or inhibit the differentiation of specific blood cell types, depending on the context of the signaling. Bone marrow niche and extrinsic signals. The bone marrow microenvironment, also known as the niche, provides essential signals for the regulation of hematopoiesis. The niche is composed of stromal cells, endothelial cells, and extracellular matrix components. HSCs interact with the niche through cell adhesion molecules and extracellular matrix proteins, such as vascular endothelial growth factor VEGF stimulates the growth of blood vessels in the bone marrow, providing oxygen and nutrients necessary for hematopoiesis. CXCL12 SDF1. This chemokine is produced by stromal cells in the niche and plays a critical role in HSC retention and migration. It binds to the CXCR4 receptor on HSCs, influencing their positioning within the niche. Regulation of lineage commitment and differentiation. Hematopoiesis involves the stepwise differentiation of HSCs into various committed progenitor cells that will give rise to specific blood cell lineages. The major lineages include erythroid lineage, controlled primarily by EPO, GATA1, and other transcription factors that promote the differentiation of erythroid progenitors into mature red blood cells. Myeloid lineage includes the formation of granulocytes, macrophages, and megakaryocytes. This process is influenced by transcription factors such as 2.1 and CEBP, as well as growth factors like GCSF and MCSF. Lymphoid lineage. Lymphoid progenitors give rise to T cells, B cells, and NK cells. This differentiation is regulated by Notch signaling, IL-7, and transcription factors like Ikaros and PU.1. Hematopoiesis in disease. Disruption of the regulatory mechanisms governing hematopoiesis can lead to a variety of hematologic diseases, such as anemia, leukemia, and other blood disorders. Hematopoietic malignancies like leukemia result from mutations in genes that regulate HSCs or progenitor cells, leading to uncontrolled proliferation of abnormal cells. Bone marrow failure syndromes, such as aplastic anemia, occur when the marrow is unable to produce adequate blood cells. Previous year questions. Which of the following cytokines is most important for the regulation of erythropoiesis? A. IL-6. B. Erythropoietin. C. Stem cell factor. D. GCSF. Answer. Previous year questions. Which of the following cytokines is most important for the regulation of erythropoiesis? A. IL-6. B. Erythropoietin. C. D. GCSF. Answer. B. Erythropoietin. CSIRNET 2018. The transcription factor PU.1 is primarily involved in the differentiation of which of the following blood cells? A. Erythrocytes. B. Neutrophils. C. T lymphocytes. D. Megakaryocytes. Answer. B. Neutrophils. CSIRNET 2017. Which of the following is the primary function of the transcription factor PU.1 is primarily involved in the differentiation of which of the following blood cells? A. Erythrocytes. B. Neutrophils. C. T lymphocytes. D. Megakaryocytes. Answer. B. Answer. B. Neutrophils. CSIRNET 2017. Which of the following is the primary? The transcription factor PU.1 is primarily involved in the differentiation of which of the following blood cells? A. Erythrocytes. B. Neutrophils. C. T lymphocytes. D. Megakaryocytes. Answer. B. Neutrophils. CSIRNET 2017. Which of the following is a primary function of thrombopoietin TPO? A. Promotion of red blood. Which of the following cytokines is most important for the regulation of erythropoiesis? A. IL-6. B. Erythropoietin. C. Stem cell factor. D. GCSF. Answer. B. Erythropoietin. CSIRNET 2018. The transcription factor PU.1 is primarily involved in the differentiation of which of the following blood cells? A. Erythrocytes. B. Neutrophils. C. T lymphocytes. D. Megakaryocytes. Answer. B. Neutrophils. CSIRNET 2017. Which of the following is a primary function of thrombopoietin TPO? A. Promotion of red blood cell production. B. Promotion of platelet production. C. Promotion of neutrophil production. D. Regulation of B cell differentiation. Answer. B. Promotion of platelet production. CSIRNET 2016. These points cover the basics to advanced aspects of cellular communication and hematopoiesis regulation. The interactions between hematopoietic stem cells, the bone marrow niche, cytokines, and transcription factors form the foundation for understanding hematopoiesis. Which of the following is a primary function of thrombopoietin TPO? A. Promotion of red blood cell production. B. Promotion of platelet production. C. Promotion of neutrophil production. D. Regulation of B cell differentiation. Answer. B. Promotion of platelet production. CSIRNET 2016. These points cover the basics to advanced aspects of cellular communication and hematopoiesis regulation. The interactions between hematopoietic stem cells, the bone marrow niche, cytokines, and transcription factors form the foundation for understanding hematopoiesis at both physiological and pathological levels. You said. General principles of cell communication. ChatGPT said. Principles of cell communication. Cell communication is an essential process for maintaining homeostasis and coordinating the functions of cells in multicellular organisms. This communication allows cells to respond to internal and external signals, regulate their behavior, and ensure proper development, immune response, and tissue repair. Cell communication can be direct or indirect, depending on the distance between the communicating cells and the nature of the signal. Key mechanisms of cell communication. Signal reception. Cells receive external signals through specific receptor molecules located on the cell surface or inside the cell. These receptors are highly selective and can recognize particular molecules, called ligands. The binding of the ligand to its receptor initiates a signal transduction cascade that leads to a cellular response. Receptors can be cell surface receptors. These are typically transmembrane proteins that bind to hydrophilic ligands, such as peptides and cytokines, which cannot cross the plasma membrane. Intracellular receptors. These receptors, located in the cytoplasm or nucleus, interact with hydrophobic ligands, such as steroid hormones, that can pass through the plasma membrane and bind to these receptors inside the cell. Signal transduction. Once a ligand binds to its receptor, the receptor undergoes a conformational change that activates intracellular signaling pathways. These pathways involve a series of molecular events where each protein or molecule in the pathway activates the next one in a cascade-like manner. Signal transduction ensures that the initial signal is amplified and transmitted across the cell. Key components in signal transduction include second messengers. Molecules like cyclic AMP, CAMP, calcium ions, Ca2, and inositol IP act as secondary messengers that relay the signal from the receptor to intracellular targets, amplifying the signal. Protein kinases. Enzymes like protein kinase A, PKA, protein kinase C, PKC, and mitogen-activated protein kinases, MAPKs, phosphorylate other proteins to activate or deactivate their functions. This phosphorylation cascade is crucial in cellular responses such as cell division, growth, and differentiation. Types of cell communication. Cell communication can be categorized based on the distance between the communicating cells. Autocrine signaling. A cell secretes a signal molecule that binds to receptors on its own surface or on neighboring cells of the same type. This type of signaling is common in immune cells and cancer cells, where a cell can regulate its own activity. Paracrine signaling. In this case, the signal is released into the extracellular fluid and acts on nearby cells. A classic example of paracrine signaling is the communication between nerve cells and their target cells at synapses, where neurotransmitters like acetylcholine are released to transmit signals. Endocrine signaling. Hormones are secreted by endocrine glands, such as the thyroid or pancreas, and travel through the bloodstream to distant target cells. An example is insulin signaling, where insulin released from the pancreas travels through the bloodstream to regulate glucose uptake in various tissues. An example is insulin signaling, where insulin released from the pancreas travels through the bloodstream to regulate glucose uptake in various tissues. Juxtacrine signaling. Direct cell-to-cell communication. In this type of signaling, cells communicate through direct contact via specialized structures like gap junctions or cadherins. This communication is essential during development and immune cell interactions. Cellular responses to signaling. Once a signal is transduced within the cell, it leads to a specific response. Responses can be varied and depend on the type of signal, the cell's receptor, and its state of differentiation. Responses include Gene expression regulation. Many signaling pathways influence the expression of specific genes, which leads to changes in protein production. For instance, the activation of the MEPK pathway can result in the transcription of genes involved in cell proliferation. Changes in cellular metabolism. Some signals, such as insulin, can alter metabolic processes in a cell, like glucose uptake or lipid synthesis. Cellular movement or migration. Chemotactic signals, such as those involved in immune responses, can direct cells to move toward or away from a stimulus. Cell death apoptosis. In certain cases, signals can initiate programmed cell death, which is essential for maintaining tissue homeostasis and eliminating damaged cells. Feedback regulation in cell communication. Cellular signaling pathways are often regulated by feedback mechanisms to maintain balance and prevent overactivation. Feedback regulation can be negative feedback, a process where the product of the signaling pathway inhibits the activity of components earlier in the pathway, thus limiting the response. For example, when cortisol levels rise in response to stress, negative feedback ensures that cortisol production is reduced once the stressor is removed. Positive feedback, a loop where the product of the pathway amplifies the initial signal, leading to a stronger or prolonged response. An example is the activation of a small number of platelets in response to a wound, which then leads to a larger recruitment of platelets in the process of clot formation. Key molecules in cell communication. Ligands. These are cell death apoptosis. In certain cases, signals can initiate programmed cell death, which is essential for maintaining tissue homeostasis and eliminating damaged cells. Feedback regulation in cell communication. Cellular signaling pathways are often regulated by feedback mechanisms to maintain balance and prevent overactivation. Feedback regulation can be negative feedback, a process where the product of the signaling pathway inhibits the activity of components earlier in the pathway, thus limiting the response. For example, when cortisol levels rise in response to stress, negative feedback ensures that cortisol production is reduced once the stressor is removed. Positive feedback, a loop where the product of the pathway amplifies the initial signal, leading to a stronger or prolonged response. An example is the activation of a small number of platelets in response to a wound, which then leads to a larger recruitment of platelets in the process of clot formation. Key molecules in cell communication. Ligands. These are signaling molecules, including hormones, neurotransmitters, growth factors, and cytokines that initiate communication between cells. Receptors. These are proteins located on the cell surface or inside the cell that bind to ligands and trigger a signal transduction cascade. G proteins. Many receptors are coupled to G proteins, which are involved in transmitting the signal from the receptor to the inside of the cell by activating second messengers. Protein phosphatases. These enzymes reverse the effects of kinases by dephosphorylating target proteins, thus regulating signal intensity and duration. Transcription factors. These are activated by signaling pathways and regulate the expression of genes in response to external signals. Previous year questions. Which of the following is a second messenger molecule commonly involved in intracellular signaling? A. Protein kinase. B. CAMP. C. Calcium ion. D. Both B and C. Answer. D. Answer. D. Both B and C. CSIRNET 2018. Which of the following signaling mechanisms involves the secretion of signaling molecules that act on the same cell that secreted them? A. Paracrine signaling. B. Endocrine signaling. C. Autocrine signaling. D. Juxtacrine signaling. Answer. C. Autocrine signaling. CSIRNET 2017. In receptor-mediated signaling, the receptor typically undergoes which of the following changes after ligand binding? A. Conformational change. B. Phosphorylation. C. Degradation. D. Synthesis of new proteins. Answer. A conformational change. CSIRNET 2016. Understanding these general principles of cell communication is crucial for a comprehensive grasp of how cellular processes are regulated in both normal and disease states. These signaling pathways allow cells to interact with their environment and respond appropriately, ensuring organismal health and function. You said. Cell adhesion and roles of different adhesion molecules. ChatGPT said. Cell adhesion and the roles of different adhesion molecules. Cell adhesion is a fundamental process that enables cells to interact with each other and with the extracellular matrix (ECM) to maintain tissue structure, function, and integrity. Adhesion molecules are specialized proteins that mediate these interactions, playing key roles in various biological processes such as tissue development, immune responses, wound healing, and cancer metastasis. These molecules allow cells to form tissues, coordinate activities, and maintain cellular organization. There are several types of adhesion molecules, and their functions are critical for regulating cell behavior and tissue homeostasis. Types of cell adhesion. Cell adhesion. This type of adhesion involves the interaction between cells, allowing them to bind to each other. It is crucial for maintaining tissue architecture and function. Cell adhesion is mediated by cadherins. Calcium-dependent adhesion molecules are the primary mediators of cell adhesion. They interact with cadherins on adjacent cells, creating strong, homophilic interactions. This interaction is regulated by calcium ions, which facilitate the binding between the cadherin extracellular domains. Immunoglobulin Ig-like adhesion molecules also mediate cell-cell interactions. They are involved in immune cell recognition and adhesion, and their function can be influenced by the binding of ligands or other signaling molecules. Cell matrix adhesion. This type of adhesion involves the interaction between cells in the extracellular matrix (ECM), a network of proteins and carbohydrates that provide structural support to tissues. The major adhesion molecules involved in cell matrix adhesion are integrins and selectins. These molecules connect the cytoskeleton of the cell to the ECM, allowing cells to sense the mechanical properties of their environment. Roles of different adhesion molecules. Cadherins. Cadherins are a family of transmembrane proteins that mediate calcium-dependent cell adhesion. They are involved in maintaining tissue integrity, regulating cell sorting, and organizing tissues during development. Cadherins are crucial for establishing and maintaining the structure of epithelial tissues. They form adherens junctions and desmosomes, which are responsible for strong mechanical connections between cells. E-cadherin is primarily expressed in epithelial cells and plays a key role in maintaining epithelial integrity and structure. N-cadherin is found in neural tissues and plays a role in neuronal development and synaptic plasticity. P-cadherin is involved in the adhesion between placental cells during pregnancy. Desmogleins and desmoglins are types of cadherins found in desmosomes, which provide mechanical strength to tissues by linking the cytoskeletons of adjacent cells. Integrins. Integrins are a family of transmembrane receptors that mediate cell matrix adhesion. Desmogleins and desmoglins are types of cadherins found in desmosomes, which provide mechanical strength to tissues by linking the cytoskeletons of adjacent cells. Integrins. Integrins are a family of transmembrane receptors that mediate cell matrix adhesion by connecting the intracellular cytoskeleton with the extracellular matrix. Integrins play vital roles in cell migration, tissue repair, immune responses, and cell survival. They are composed of alpha and beta subunits, which determine the specificity of binding to different ECM ligands such as fibronectin, collagen, and laminin. Integrins participate in signaling pathways that regulate cellular responses to the environment, including growth, differentiation, and apoptosis. Alpha-5 beta-1 integrin binds to fibronectin and is involved in cell migration and wound healing. Alpha-1 beta-1 integrin interacts with collagen and is essential for maintaining the structural integrity of tissues. Alpha versus beta-3 integrin binds to multiple ECM components and is involved in angiogenesis, formation of new blood vessels, and tumor progression. Alpha-L beta-2 integrin LFA1 is essential for immune cell adhesion, particularly in leukocyte trafficking and immune responses. Selectins. Selectins are a group of adhesion molecules that mediate transient interactions between cells, particularly during immune responses. They are involved in the rolling of leukocytes, white blood cells, along the blood vessel walls, a key step in the immune response. Selectins recognize specific carbohydrate structures on glycoproteins and glycolipids on the surface of other cells. P-selectin is expressed on endothelial cells during inflammation and mediates the adhesion of leukocytes to the vessel wall. P-selectin is stored in platelets and endothelial cells and is rapidly expressed at the site of injury to mediate platelet aggregation and leukocyte recruitment. L-selectin is expressed on leukocytes and plays a role in their trafficking to lymph nodes and sites of inflammation. Immunoglobulin superfamily adhesion molecules. The immunoglobulin Ig superfamily includes a variety of cell adhesion molecules that mediate both cell and cell matrix interactions, particularly in the immune system. These molecules are involved in immune cell signaling, antigen presentation, and tissue repair. They can be either cell surface proteins or secreted factors that interact with receptors on other cells. ICAM1 intercellular adhesion molecule 1 is expressed on endothelial cells and binds to integrins on leukocytes, facilitating ICAM1 intercellular adhesion molecule 1 is expressed on endothelial cells and binds to integrins on leukocytes, facilitating their migration to sites of infection or injury. VCAM1 vascular cell adhesion molecule 1 also mediates the adhesion of leukocytes to the vascular endothelium and is involved in inflammation and immune responses. NCAM neural cell adhesion molecule is involved in neuronal adhesion and synaptic plasticity, contributing to brain development and learning. Connexins gap junctions. Gap junctions are specialized cell junctions that allow direct communication between adjacent cells. They are formed by connexin proteins that create channels for the exchange of ions, small molecules, and signaling compounds. Gap junctions are critical for maintaining tissue homeostasis and facilitating coordinated cellular responses, particularly in cardiac and smooth muscle cells where synchronized contraction is necessary. Connexins play a role in signal transduction and metabolic cooperation between cells. Cadherin catene. Gap junctions are critical for maintaining tissue homeostasis and facilitating coordinated cellular responses, particularly in cardiac and smooth muscle cells where synchronized contraction is necessary. Connexins play a role in signal transduction and metabolic cooperation between cells. Cadherin catene complex. The cadherin catene complex is a group of intracellular proteins that link cadherins to the actin cytoskeleton. This complex is essential for stabilizing cadherin-mediated cell junctions. Catenins such as beta-catenin and P12O-catenin interact with the cytoplasmic domain of cadherins and help to stabilize cell adhesion, regulate cell signaling, and promote cell survival. Beta-catenin, in particular, is involved in the WNT signaling pathway and can influence gene expression related to cell growth and differentiation. Roles of adhesion molecules in development and disease. Development. During embryonic development, cell adhesion molecules are crucial for tissue morphogenesis, the formation of organ systems, and cell differentiation. The proper regulation of cadherins, integrins, and selectins ensures that cells adhere to the right structures and communicate effectively to drive developmental processes. Immune response. In immune system function, adhesion molecules regulate the movement of immune cells to sites of infection or injury. Integrins and selectins mediate the rolling, adhesion, and transmigration of leukocytes through the endothelial cells of blood vessels, facilitating immune surveillance and inflammation. Cancer metastasis. In cancer, the loss or dysregulation of adhesion molecules can lead to the detachment of tumor cells from the primary site, promoting metastasis. For instance, decreased expression of E-cadherin is often observed in epithelial cancers, allowing tumor cells to break away and spread to other tissues. Wound healing and tissue repair. During wound healing, integrins and cadherins coordinate the migration and proliferation of cells at the injury site. These molecules also contribute to the formation of new blood vessels and tissue remodeling. Previous year questions. Which of the following adhesion molecules is primarily involved in the formation of adherens junctions? A. Integrins. B. Cadherins. B. Which of the following adhesion molecules is primarily involved in the formation of adherens junctions? A. Integrins. B. Cadherins. C. Selectins. D. Connexins. Answer. B. Cadherins. CSIRNET 2018. Which of the following adhesion molecules is involved in leukocyte adhesion during immune responses? A. ICAM-1. B. E-cadherin. C. Fibronectin. D. Laminin. Answer. A. ICAM-1. CSIRNET 2017. What is the primary role of integrins in cell adhesion? A. Binding of cadherins to the cytoskeleton. B. Mediating cell adhesion. C. Mediating cell matrix adhesion. D. Promoting gap junction formation. Answer. C. Mediating cell matrix adhesion. CSIRNET 2016. Understanding the roles of different adhesion molecules and how they regulate cellular interactions is fundamental for understanding cell behavior in both normal and disease states. These adhesion molecules are critical for processes such as tissue development, immune response, cancer metastasis, and wound healing. You said, gap junctions. ChatGPT said, gap junctions. Gap junctions are specialized intercellular channels that allow direct communication between adjacent cells. They are crucial for maintaining cellular homeostasis and coordinating the activities of cells in various tissues, such as the heart, brain, liver, and smooth muscle. Gap junctions enable the exchange of ions, small molecules, and signaling compounds, allowing cells to function in a synchronized manner. Gap junctions are formed by proteins known as connexins, which assemble to create channels that bridge the gap between adjacent cells. These channels facilitate the passage of ions, metabolites, and second messengers, thus enabling cells to communicate rapidly and efficiently. This type of communication is essential for coordinating processes like contraction in cardiac and smooth muscle, the regulation of neuronal activity, and maintaining metabolic balance across tissues. Structure of gap junctions. Gap junctions are composed of connexins, which are transmembrane proteins that oligomerize to form a channel called a connexon. A connexon is a hemachannel made up of 6 connexin subunits. 2 connexons, one from each adjacent cell, align and dock together to form a complete channel that spans the intercellular space. The size of the gap junction channel is typically 1.52 nanometers in diameter, allowing the passage of small molecules such as ions, nucleotides, and metabolites, but blocking the movement of larger molecules like proteins and RNA. Each connexin protein has a distinct molecular weight, and there are multiple types of connexins, e.g., CX43, CX32, CX26, CX40 expressed in different tissues. The specific combination of connexins in a connexon determines the properties of the gap junction, including its permeability and selectivity. Functions of gap junctions. Electrical coupling. In excitable tissues like the heart and brain, gap junctions enable the rapid transmission of electrical signals. For example, in cardiac muscle cells, cardiomyocytes, gap junctions allow the flow of ions between cells, enabling synchronized contraction. The rapid conduction of action potentials through the heart muscle is critical for maintaining coordinated heartbeats. Similarly, in smooth muscle cells, gap junctions facilitate coordinated contraction, which is important for processes such as peristalsis in the intestines. Metabolic coupling. Gap junctions also play a role in metabolic coupling by allowing the transfer of small metabolites between cells. For instance, in the liver, gap junctions allow the exchange of metabolites such as glucose, ATP, and other small signaling molecules, helping cells in a tissue coordinate metabolic processes. Signal transmission and coordination. Gap junctions facilitate the transfer of second messengers and ions, enabling the coordination of cellular responses to external signals. In tissues like the retina or cochlea, gap junctions help coordinate the response to sensory stimuli. They also enable the spread of calcium waves in non-excitable cells, helping in processes like wound healing and the regulation of cellular activities during development. Homeostasis and tissue coordination. Gap junctions are essential for maintaining tissue homeostasis by enabling the exchange of ions and small molecules, which functions of gap junctions. Electrical coupling. In excitable tissues like the heart and brain, gap junctions enable the rapid transmission of electrical signals. For example, in cardiac muscle cells, cardiomyocytes, gap junctions allow the flow of ions between cells, enabling synchronized contraction. The rapid conduction of action potentials through the heart muscle is critical for maintaining coordinated heartbeats. Similarly, in smooth muscle cells, gap junctions facilitate coordinated contraction, which is important for processes such as peristalsis in the intestines. Metabolic coupling. Gap junctions also play a role in metabolic coupling by allowing the transfer of small metabolites between cells. For instance, in the liver, gap junctions allow the exchange of metabolites such as glucose, ATP, and other small signaling molecules, helping cells in a tissue coordinate metabolic processes. Signal transmission and coordination. Gap junctions facilitate the transfer of second messengers and ions, enabling the coordination of cellular responses to external signals. In tissues like the retina or cochlea, gap junctions help coordinate the response to sensory stimuli. They also enable the spread of calcium waves in non-excitable cells, helping in processes like wound healing and the regulation of cellular activities during development. Homeostasis and tissue coordination. Gap junctions are essential for maintaining tissue homeostasis by enabling the exchange of ions and small molecules, which helps balance the internal environment of cells. This function is particularly important in epithelial and endothelial tissues, where cells need to work. This function is particularly important in epithelial and endothelial tissues, where cells need to work in unison to maintain tissue integrity and respond to environmental changes. Development and differentiation. Gap junctions play a crucial role during embryonic development and cell differentiation. They are involved in processes like cell migration, tissue patterning, and the coordination of cellular behaviors during tissue formation. The proper functioning of gap junctions is essential for the development of various organs, including the brain, heart, and lungs. Wound healing. Gap junctions are involved in wound healing, particularly during the early stages when cells at the wound site need to communicate to coordinate their repair activities. They facilitate the spread of signals, such as calcium waves, that promote cell proliferation, migration, and the synthesis of extracellular matrix proteins, which are essential for tissue repair. Regulation of gap junctions. They facilitate the spread of signals, such as calcium waves, that promote cell proliferation, migration, and the synthesis of extracellular matrix proteins, which are essential for tissue repair. Regulation of gap junctions. Gap junction communication is tightly regulated to ensure proper cellular function. Several mechanisms control the opening and closing of gap junction channels, including phosphorylation. The phosphorylation of connexins, particularly by kinases such as protein kinase C, PKC, can regulate the permeability of gap junctions. Phosphorylation can either enhance or inhibit gap junction communication depending on the type of connexin and the cellular context. Calcium. High intracellular calcium levels can induce the closure of gap junctions. This mechanism is particularly important in regulating gap junction communication in response to cellular stress, injury, or during specific physiological events like muscle contraction. pH and metabolites. Changes in intracellular pH and the concentration of certain metabolites can also influence gap junction permeability. For example, an acidic environment can lead to the closure of gap junctions, which helps to protect cells from damage. Endocytosis. Gap junctions can be internalized into the cell through a process called endocytosis. This can lead to a reduction in gap junction communication, often as a response to signals that require a change in cell-cell communication. Diseases associated with gap junctions. Defects in gap junctions and connexin function are linked to various diseases, including cardiovascular diseases. Mutations in connexins, such as CX43, can lead to arrhythmias and other heart conditions. For example, CX43 mutations have been associated with congenital heart defects and abnormal heart rhythms, arrhythmias, which can impair the heart's ability to pump blood efficiently. Neurodegenerative diseases. Abnormal gap junction communication in the brain can contribute to neurodegenerative diseases such as Alzheimer's disease. Disrupted neuronal communication can interfere with synaptic plasticity and cognitive function, which are essential for memory and learning. Skin disorders. Defects in CX26 are associated with skin disorders such as keratitis ichthyosis deafness KID syndrome. Mutations in connexin genes can disrupt cell-cell communication in the epidermis, leading to skin thickening, eye abnormalities, and hearing loss. Cancer. In many cancers, gap junction communication is impaired. Loss of connexin expression or altered gap junction function can facilitate uncontrolled cell proliferation and tumor progression. Gap junctions help maintain normal cellular organization and tissue structure, and their loss can contribute to the metastatic potential of cancer cells. Previous year questions. Which of the following proteins form the structural basis of gap junctions? A. Cadherins. B. Connexins. C. Integrins. D. Selectins. Answer. B. Connexins. CSIRNET 2018. What is the primary function of gap junctions in excitable tissues like the heart and brain? A. Mediating cell-cell adhesion. B. Facilitating rapid electrical coupling between cells. C. Connecting cells to the extracellular matrix. D. Regulating gene expression. Answer. B. Facilitating rapid electrical coupling between cells. CSIRNET 2017. Which of the following conditions is associated with mutations in connexin proteins? A. Cystic fibrosis. B. Keratitis ichthyosis deafness KID syndrome. C. Duchenne muscular dystrophy. D. Huntington's disease. Answer. B. Keratitis ichthyosis deafness KID syndrome. CSIRNET 2016. Gap junctions are crucial for maintaining tissue function and ensuring cellular communication. Their role in synchronizing cellular responses and coordinating activities across tissues underscores their importance in both normal physiology and disease states. You said. Extracellular matrix. ChatGPT said. Extracellular matrix ECM. The extracellular matrix ECM is a complex network of proteins and polysaccharides secreted by cells into the extracellular space, providing which of the following proteins form the structural basis of gap junctions? A. Cadherins. B. Connexins. C. Integrins. D. Selectins. Answer. B. Connexins. CSIRNET 2018. What is the primary function of gap junctions in excitable tissues like the heart and brain? A. Mediating cell-cell adhesion. B. Facilitating rapid electrical coupling between cells. C. Connecting cells to the extracellular matrix. D. Regulating gene expression. Answer. B. Facilitating rapid electrical coupling between cells. CSIRNET 2017. Which of the following conditions is associated with mutations in connexin proteins? A. Cystic fibrosis. B. Keratitis ichthyosis deafness KID syndrome. C. Duchenne muscular dystrophy. D. Huntington's disease. Answer. B. Keratitis ichthyosis deafness KID syndrome. CSIRNET 2016. Gap junctions are crucial for maintaining tissue function and ensuring cellular communication. Their role in synchronizing cellular responses and coordinating activities across tissues underscores their importance in both normal physiology and disease states. You said. Extracellular matrix. ChatGPT said. Extracellular matrix ECM. The extracellular matrix ECM is a complex network of proteins and polysaccharides secreted by cells into the extracellular space, providing structural and biochemical support to the surrounding cells. It is a critical component of tissue architecture, and its composition and organization influence many cellular processes, including differentiation, migration, growth, and survival. The ECM serves as a scaffold for cells and tissues, contributing to their mechanical strength, elasticity, and regulation of cellular behavior. Structure of ECM. The ECM is composed of various macromolecules, which can be broadly categorized into fibrous proteins, glycoproteins, and proteoglycans. These components interact to form a dynamic structure that adapts to the needs of the tissue. Fibrous proteins. Collagen. Collagen is the most abundant protein in the ECM and provides structural support and tensile strength to tissues. There are several types of collagen, e.g., type 1, 2, 3, IV, each found in different tissues. Type 1 collagen, for instance, is present in connective tissues like bone, skin, and tendons, while type 4 collagen is found in the basement membrane. Elastin. Elastin is a key protein in the ECM of tissues that require elasticity, such as skin, lungs, and blood vessels. It provides tissues with the ability to stretch and return to their original shape. Fibronectin. Fibronectin is a glycoprotein that plays a crucial role in cell adhesion, migration, and wound healing. It binds to integrins on the cell surface and to collagen and other ECM components. Proteoglycans. Proteoglycans are large molecules consisting of a core protein and one or more glycosaminoglycan GAG chains. These molecules have a high water content and contribute to the ECM's ability to resist compression. An example is agrecan, which is abundant in cartilage and plays a key role in maintaining tissue hydration and elasticity. Glycosaminoglycans GAGs are long, unbranched polysaccharide chains that are highly negatively charged due to sulfate and carboxyl groups. They are often attached to core proteins to form proteoglycans. The most common GAGs include hyaluronan, hyaluronic acid, chondroitin sulfate, heparin sulfate, and choroitin sulfate. These molecules play a crucial role in maintaining ECM hydration and structure. Basement membrane. The basement membrane is a specialized form of ECM that forms a thin, dense sheet-like structure at the interface between epithelial and endothelial cells in the underlying connective tissue. It is composed of specialized ECM proteins such as laminin, type 4 collagen, and neogen. The basement membrane is essential for cell adhesion, differentiation, and tissue compartmentalization. Functions of ECM. Structural support. The ECM provides a physical scaffold for tissues and organs, allowing them to maintain their shape and resist mechanical stresses. Collagen fibers are particularly important in providing tensile strength, while elastin fibers allow tissues to stretch and recoil. The ECM also helps organize cells into tissues and organs. Cell adhesion and migration. ECM components, such as fibronectin, laminin, and integrins, mediate cell adhesion to the matrix and to one another. This process is essential for tissue development, wound healing, and immune responses. The ECM also regulates cell migration, which is crucial during development, tissue repair, and metastasis. Regulation of cell behavior. The ECM has a critical role in regulating cell functions such as differentiation, proliferation, and survival. Signals from the ECM are received by cell surface receptors, mainly integrins, which transduce signals into the cell. These signals can modulate gene expression and influence cellular responses to environmental cues. Mechanical properties. The ECM provides tissues with mechanical properties such as strength, elasticity, and resilience. For instance, in bones and tendons, the ECM provides structural rigidity and tensile strength, while in tissues like cartilage, it helps resist compressive forces. Tissue development and maintenance. During embryogenesis and tissue development, the ECM guides cell migration, differentiation, and patterning. Structural support. The ECM provides a physical scaffold for tissues and organs, allowing them to maintain their shape and resist mechanical stresses. Collagen fibers are particularly important in providing tensile strength, while elastin fibers allow tissues to stretch and recoil. The ECM also helps organize cells into tissues and organs. Cell adhesion and migration. ECM components, such as fibronectin, laminin, and integrins, mediate cell adhesion to the matrix and to one another. This process is essential for tissue development, wound healing, and immune responses. The ECM also regulates cell migration, which is crucial during development, tissue repair, and metastasis. Regulation of cell behavior. The ECM has a critical role in regulating cell functions such as differentiation, proliferation, and survival. Signals from the ECM are received by cell surface receptors, mainly integrins, which transduce signals into the cell. These signals can modulate gene expression and influence cellular responses to environmental cues. Mechanical properties. The ECM provides tissues with mechanical properties such as strength, elasticity, and resilience. For instance, in bones and tendons, the ECM provides structural rigidity and tensile strength, while in tissues like cartilage, it helps resist compressive forces. Tissue development and maintenance. During embryogenesis and tissue development, the ECM guides cell migration, differentiation, and patterning. It helps establish tissue-specific structures and enables the proper organization of cells. In mature tissues, the ECM supports the maintenance of tissue architecture and function. Wound healing and tissue repair. The ECM plays a central role in wound healing by providing a scaffold for the migration and proliferation of cells such as fibroblasts, endothelial cells, and epithelial cells. It also serves as a reservoir for growth factors that stimulate repair processes. For example, fibronectin and collagen are involved in tissue remodeling during wound healing. Signaling and cellular communication. The ECM influences cellular behavior through mechanical and biochemical signals. The interaction between integrins on the cell surface and ECM components can trigger intracellular signaling pathways that regulate processes such as gene expression, cytoskeletal rearrangement, and cell survival. This signaling is vital for processes like tissue homeostasis, immune response, and cancer progression. ECM and disease. Cancer. The ECM is involved in cancer progression by affecting tumor cell behavior and metastasis. Tumor cells often produce altered ECM components, which help them invade surrounding tissues and migrate to distant sites. Changes in ECM stiffness and composition can also influence the behavior of cancer cells and promote their growth and survival. Fibrosis. Fibrosis refers to the excessive accumulation of ECM components, particularly collagen, which leads to tissue scarring and impaired organ function. It can occur in various organs, including the lungs, pulmonary fibrosis, liver cirrhosis, and kidneys, renal fibrosis. Fibrosis disrupts normal tissue architecture and impairs organ function. Osteoarthritis. In osteoarthritis, the degradation of ECM components in cartilage, particularly agrecan and type 2 collagen, leads to the breakdown of joint function. This results in pain, inflammation, and reduced mobility. Ehlers-Danlos syndrome. This group of disorders is caused by defects in collagen synthesis or processing, leading The extracellular matrix (ECM) is a complex network of proteins and polysaccharides secreted by cells into the extracellular space, providing structural and biochemical support to the surrounding cells. It is a critical component of tissue architecture, and its composition and organization influence many cellular processes, including differentiation, migration, growth, and survival. The ECM serves as a scaffold for cells and tissues, contributing to their mechanical strength, elasticity, and regulation of cellular behavior. Structure of ECM. The ECM is composed of various macromolecules, which can be broadly categorized into fibrous proteins, glycoproteins, and proteoglycans. These components interact to form a dynamic structure that adapts to the needs of the tissue. Fibrous proteins. Collagen. Collagen is the most abundant protein in the ECM and provides structural support and tensile strength to tissues. There are several types of collagen, e.g., type 1, 2, 3, IV, each found in different tissues. Type 1 collagen, for instance, is present in connective tissues like bone, skin, and tendons, while type 4 collagen is found in the basement membrane. Elastin. Elastin is a key protein in the ECM of tissues that require elasticity, such as skin, lungs, and blood vessels. It provides tissues with the ability to stretch and return to their original shape. Fibronectin. Fibronectin is a glycoprotein that plays a crucial role in cell adhesion, migration, and wound healing. It binds to integrins on the cell surface and to collagen and other ECM components. Proteoglycans. Proteoglycans are large molecules consisting of a core protein and one or more glycosaminoglycan GAG chains. These molecules have a high water content and contribute to the ECM's ability to resist compression. An example is agrecan, which is abundant in cartilage and plays a key role in maintaining tissue hydration and elasticity. Glycosaminoglycans GAGs are long, unbranched polysaccharide chains that are highly negatively charged due to sulfate and carboxyl groups. They are often attached to core proteins to form proteoglycans. The most common GAGs include hyaluronan, hyaluronic acid, chondroitin sulfate, heparin sulfate, and choroitin sulfate. These molecules play a crucial role in maintaining ECM hydration and structure. Basement membrane. The basement membrane is a specialized form of ECM that forms a thin, dense sheet. The ECM is composed of various macromolecules, which can be broadly categorized into fibrous proteins, glycoproteins, and proteoglycans. These components interact to form a dynamic structure that adapts to the needs of the tissue. Fibrous proteins. Collagen. Collagen is the most abundant protein in the ECM and provides structural support and tensile strength to tissues. There are several types of collagen, e.g., type 1, 2, 3, IV, each found in different tissues. Type 1 collagen, for instance, is present in connective tissues like bone, skin, and tendons, while type 4 collagen is found in the basement membrane. Elastin. Elastin is a key protein in the ECM of tissues that require elasticity, such as skin, lungs, and blood vessels. It provides tissues with the ability to stretch and return to their original shape. Fibronectin. Fibronectin is a glycoprotein that plays a crucial role in cell adhesion, migration, and wound healing. It binds to integrins on the cell surface and to collagen and other ECM components. Proteoglycans. Proteoglycans are large molecules consisting of a core protein and one or more glycosaminoglycan GAG chains. These molecules have a high water content and contribute to the ECM's ability to resist compression. An example is agrecan, which is abundant in cartilage and plays a key role in maintaining tissue hydration and elasticity. Glycosaminoglycans GAGs are long, unbranched polysaccharide chains that are highly negatively charged due to sulfate and carboxyl groups. They are often attached to core proteins to form proteoglycans. The most common GAGs include hyaluronan, hyaluronic acid, chondroitin sulfate, heparan sulfate, and choroitin sulfate. These molecules play a crucial role in maintaining ECM hydration and structure. Basement membrane. The basement membrane is a specialized form of ECM that forms a thin, dense sheet-like structure at the interface between epithelial and endothelial cells in the underlying connective tissue. It is composed of specialized ECM proteins such as laminin, type 4 collagen, and ketogen. The basement membrane is essential for cell adhesion, differentiation, and tissue compartmentalization. Functions of ECM. Structural support. The ECM provides a physical scaffold for tissues and organs, allowing them to maintain their shape and resist mechanical stresses. Collagen fibers are particularly important in providing tensile strength, while elastin fibers allow tissues to stretch and recoil. The ECM also helps organize cells into tissues and organs. Cell adhesion and migration. ECM components, such as fibronectin, laminin, and integrins, mediate cell adhesion to the matrix and to one another. This process is essential for tissue development, wound healing, and immune responses. The ECM also regulates cell migration, which is crucial during development, tissue repair, and metastasis. Regulation of cell behavior. The ECM has a critical role in regulating cell functions such as differentiation, proliferation, and survival. Signals from the ECM are received by cell surface receptors, mainly integrins, which transduce signals into the cell. These signals can modulate gene expression and influence cellular responses to environmental cues. Mechanical properties. The ECM provides tissues with mechanical properties such as strength, elasticity, and resilience. For instance, in bones and tendons, the ECM provides structural rigidity and tensile strength, while in tissues like cartilage, it helps resist compressive forces. Tissue development and maintenance. During embryogenesis and tissue development, the ECM guides cell migration, differentiation, and patterning. It helps establish tissue-specific structures and enables the proper organization of cells. In mature tissues, the ECM supports the maintenance of tissue architecture and function. Wound healing and tissue repair. The ECM plays a central role in wound healing by providing a scaffold for the migration and proliferation of cells such as fibroblasts, endothelial cells, and epithelial cells. It also serves as a reservoir for growth factors that stimulate repair processes. For example, fibronectin and collagen are involved in tissue remodeling during wound healing. Signaling and cellular communication. The ECM influences cellular behavior through mechanical and biochemical signals. The interaction between integrins on the cell surface and ECM components can trigger intracellular signaling pathways that regulate processes such as gene expression, cytoskeletal rearrangement, and cell survival. This signaling is vital for processes like tissue homeostasis, immune response, and cancer progression. ECM and disease. Cancer. The ECM is involved in cancer progression by affecting tumor cell behavior and metastasis. Tumor cells often produce altered ECM components, which help them invade surrounding tissues and migrate to distant sites. Changes in ECM stiffness and composition can also influence the behavior of cancer cells and promote their growth and survival. Fibrosis. Fibrosis refers to the excessive accumulation of ECM components, particularly collagen, which leads to tissue scarring and impaired organ function. It can occur in various organs, including the lungs, pulmonary fibrosis, liver cirrhosis, and kidneys, renal fibrosis. Fibrosis disrupts normal tissue architecture and impairs organ function. Osteoarthritis. In osteoarthritis, the degradation of ECM components in cartilage, particularly agrecan and type 2 collagen, leads to the breakdown of joint function. This results in pain, inflammation, and reduced mobility. Ehlers-Danlos syndrome. This group of disorders is caused by defects in collagen synthesis or processing, leading to fragile skin, hyperelastic joints, and a tendency to develop scars and bruises easily. It highlights the importance of ECM integrity in maintaining tissue strength and flexibility. Marfan syndrome. Marfan syndrome is a connective tissue disorder caused by mutations in the gene encoding fibrillin-1, a glycoprotein that is essential for ECM organization, particularly in elastic tissues. This leads to cardiovascular, skeletal, and ocular abnormalities. Regulation of ECM remodeling. The ECM is not a static structure but a dynamic one that undergoes constant remodeling, which is crucial for tissue development, maintenance, and repair. This remodeling is controlled by several enzymes, including matrix metalloproteinases (MMPs). MMPs are a family of enzymes that degrade various ECM components. They are involved in tissue remodeling, wound healing, and cell migration. MMP activity is tightly regulated by tissue inhibitors of metalloproteinases (TIMPs) to prevent uncontrolled ECM degradation. Integrins and focal adhesions. Integrins are transmembrane receptors that connect the ECM to the actin cytoskeleton. They are involved in ECM remodeling by mediating cellular forces that influence the mechanical properties of the matrix. Focal adhesions, which are complexes of integrins and intracellular signaling proteins, play a critical role in regulating ECM remodeling. Growth factors. Growth factors such as transforming growth factor beta (TGF-beta), fibroblast growth factor (FGF), and vascular endothelial growth factor (VEGF) regulate ECM production and remodeling. These growth factors bind to specific receptors on the cell surface, initiating intracellular signaling pathways that promote ECM synthesis or degradation. Previous year questions. Which of the following ECM components are primarily responsible for the tensile strength of tissues? A. Elastin. B. Collagen. C. Fibronectin. D. Proteoglycans. Answer. B. Collagen. CSIRNET 2018. What is the role of integrins in ECM? A. Degrade ECM components. B. Act as receptors for growth factors. C. Mediate cell adhesion and signal transduction. D. Store ECM components. Answer. C. Mediate cell adhesion and signal transduction. CSIRNET 2017. Which protein is primarily involved in the elasticity of the ECM? A. Collagen. B. Elastin. C. Fibronectin. D. Hyaluronan. Answer. B. Elastin. CSIRNET 2016. The extracellular matrix is an essential component in tissue structure and function. Its dynamic nature allows cells to interact with their environment, respond to mechanical and biochemical signals, and maintain tissue integrity across various biological processes. Understanding the ECM is crucial for studying development, disease progression, and therapeutic strategies for conditions involving ECM remodeling. You said, integrins. ChatGPT said, integrins. Integrins are a large family of transmembrane glycoproteins that play a crucial role in mediating cell adhesion to the extracellular matrix (ECM) and to other cells. They are also involved in cell signaling and regulating various cellular functions such as migration, proliferation, differentiation, and survival. Integrins form an essential link between the extracellular environment and the intracellular signaling machinery, allowing cells to respond to mechanical and biochemical cues. Structure of integrins. Integrins are heterodimeric proteins, meaning they are composed of two subunits, an alpha-alpha subunit and a beta-beta subunit. Each integrin heterodimer has a distinct combination of alpha and beta subunits, and different combinations are expressed on different cell types. There are at least 18 alpha and 8 beta subunits, leading to the formation of 24 distinct integrin receptors. The subunits are non-covalently associated, and the extracellular domains of integrins mediate binding to ECM components, e.g., fibronectin, collagen, laminin, while the intracellular domains interact with the cytoskeleton, mainly actin filaments, through linker proteins. Alpha subunits. The alpha subunits determine the ligand binding specificity of the integrin. They are involved in binding to ECM components such as collagen, fibronectin, and laminin. Beta subunits. The beta subunits are involved in intracellular signaling and cytoskeletal interactions. The type of beta subunit in the integrin determines the downstream signaling pathways that the integrin activates. Mechanism of integrin function. Cell adhesion. Integrins mediate cell adhesion to the ECM by binding to specific ligands in the matrix. When integrins interact with ECM components such as fibronectin, collagen, or laminin, they trigger a conformational change in the integrin, which is essential for strong attachment between the cell and the matrix. Signal transduction. Upon ECM binding, integrins transduce signals from the extracellular environment into the cell. These signals are important for regulating cellular behavior, such as survival, migration, differentiation, and gene expression. Integrins can initiate inside-out signaling, which activates the integrin itself, and outside-in signaling, which transduces extracellular signals into the cell. Inside-out signaling occurs when intracellular signals lead to conformational changes in integrins, increasing their affinity for ECM ligands. This is crucial during cell migration and activation. Outside-in signaling involves the transmission of signals from the ECM to the cell, influencing cellular responses like adhesion strengthening, cytoskeletal rearrangement, and gene expression. Focal adhesions. Integrins cluster into large complexes known as focal adhesions. These are dynamic structures that anchor the cell to the ECM and connect integrins to the actin cytoskeleton inside the cell. Focal adhesions contain several proteins, such as talin, vinculin, paxillin, and filamin, which play a role in signal transduction and the regulation of cell movement. The assembly and disassembly of focal adhesions are tightly regulated and influence cellular processes like migration and proliferation. Integrins are heterodimeric proteins, meaning they are composed of two subunits, an alpha-alpha subunit and a beta-beta subunit. Each integrin heterodimer has a distinct combination of alpha and beta subunits, and different combinations are expressed on different cell types. There are at least 18 alpha and 8 beta subunits, leading to the formation of 24 distinct integrin receptors. The subunits are non-covalently associated, and the extracellular domains of integrins mediate binding to ECM components, e.g., fibronectin, collagen, laminin, while the intracellular domains interact with the cytoskeleton, mainly actin filaments, through linker proteins. Alpha subunits. The alpha subunits determine the ligand binding specificity of the integrin. They are involved in binding to ECM components such as collagen, fibronectin, and laminin. Beta subunits. The beta subunits are involved in intracellular signaling and cytoskeletal interactions. The type of beta subunit in the integrin determines the downstream signaling pathways that the integrin activates. Mechanism of integrin function. Cell adhesion. Integrins mediate cell adhesion to the ECM by binding to specific ligands in the matrix. When integrins interact with ECM components such as fibronectin, collagen, or laminin, they trigger a conformational change in the integrin, which is essential for strong attachment between the cell and the matrix. Signal transduction. Upon ECM binding, integrins transduce signals from the extracellular environment into the cell. These signals are important for regulating cellular behavior, such as survival, migration, differentiation, and gene expression. Integrins can initiate inside-out signaling, which activates the integrin itself, and outside-in signaling, which transduces extracellular signals into the cell. Inside-out signaling occurs when intracellular signals lead to conformational changes in integrins, increasing their affinity for ECM ligands. This is crucial during cell migration and activation. Outside-in signaling involves the transmission of signals from the ECM to the cell, influencing cellular responses like adhesion strengthening, cytoskeletal rearrangement, and gene expression. Focal adhesions. Integrins cluster into large complexes known as focal adhesions. These are dynamic structures that anchor the cell to the ECM and connect integrins to the actin cytoskeleton inside the cell. Focal adhesions contain several proteins, such as talin, vinculin, paxillin, and filamin, which play a role in signal transduction and the regulation of cell movement. The assembly and disassembly of focal adhesions are tightly regulated and influence cellular processes like migration and proliferation. Types of integrins and their ligands. Integrins are classified based on the combination of alpha and beta subunits. Some key integrins and their ligands include alpha-1 beta-1 binds to collagen. It is important for cell attachment to the ECM and is involved in tissue organization. Alpha-2 beta-1 binds to collagen and laminin. It plays a role in cell migration and interaction with the ECM in various tissues. Alpha-3 beta-1 binds to laminin. It is crucial for epithelial cell adhesion to the basement membrane. Alpha-5 beta-1 binds to fibronectin. This integrin is involved in cell migration, wound healing, and angiogenesis. Alpha versus beta-3 binds to fibronectin, and collagen. This integrin is involved in angiogenesis, wound healing, and platelet aggregation. Alpha-L beta-2 LFA1 binds to ICAMs, intercellular adhesion molecules. It is crucial for immune cell interactions, such as those between T cells and endothelial cells during immune responses. Functions of integrins. Cell adhesion and migration. Integrins play a central role in cell adhesion to the ECM, which is a prerequisite for processes such as tissue development, wound healing, and immune responses. By binding to ECM components, integrins provide cells with a means of anchorage, preventing detachment. Additionally, integrins are involved in cell migration, where they regulate focal adhesions and control the cytoskeleton's ability to contract and extend, facilitating movement through the ECM. Signal transduction. Integrins are critical for signal transduction. Through interactions with proteins in the cytoplasm, they help activate various signaling pathways, such as the MEPK mitogen-activated protein kinase pathway, the PI3K bisphoinositide 3 kinase pathway, and the Rho family GTPases pathway. These pathways are involved in regulating gene expression, cell survival, and the cytoskeletal dynamics required for migration. Tissue development and maintenance. Integrins play a significant role in tissue formation and maintenance by regulating the interactions between cells and the ECM during development. For example, integrins contribute to the formation of the basement membrane and epithelial tissues and mediate interactions between endothelial cells and the ECM in blood vessel formation angiogenesis. Wound healing. During wound healing, integrins regulate cell migration, proliferation, and differentiation at the injury site. Integrin-mediated adhesion to Integrin-mediated adhesion to ECM components like fibronectin and collagen helps to rebuild the damaged tissue. Integrins are also involved in the remodeling of the ECM at the site of injury. Immune response. Integrins are essential for immune cell function. For instance, LFA1 alpha L beta-2 integrin is crucial for immune cell adhesion to For instance, LFA1 alpha L beta-2 integrin is crucial for immune cell adhesion to endothelial cells during inflammation and for T cell activation. This interaction facilitates the migration of immune cells to sites of infection or injury. LFA alpha L beta-2 integrin is crucial for immune cell adhesion to endothelial cells during inflammation and for T cell activation. This interaction facilitates the migration of immune cells to sites of infection or injury. Cellular differentiation and survival. Integrins are involved in the regulation of cell differentiation and survival. For example, during development, integrins help guide stem cells to differentiate into specific lineages. They also regulate survival signals by activating focal adhesion kinase FAK and other kinases, which help protect cells from apoptosis, programmed cell death. Pathological implications of integrins. Cancer metastasis. Integrins play a pivotal role in cancer progression and metastasis. The ability of tumor cells to invade surrounding tissues and migrate to distant sites is partially controlled by integrin interactions with the ECM. Altered integrin signaling can facilitate tumor cell detachment from the primary tumor, migration through the ECM, and establishment of secondary tumors in distant organs. Inflammatory diseases. Integrins, especially beta-2 integrins, are involved in leukocyte adhesion and migration during inflammation. Dysregulation of integrin function can result in excessive inflammation and tissue damage. In diseases such as rheumatoid arthritis, integrin inhibitors are being studied as therapeutic agents to reduce inflammation. Platelet aggregation and thrombosis. Alpha IIb beta-3 integrin is critical in platelet aggregation during blood clotting. Dysfunction or overactivation of alpha IIb beta-3 can lead to thrombus formation, contributing to Dysfunction or overactivation of alpha IIb beta-3 can lead to thrombus formation, contributing to conditions like heart attacks or strokes. Developmental disorders. Integrins are essential for normal development, and mutations in integrin genes can lead to congenital defects. For example, integrin alpha 6 beta 4 mutations cause epidermolysis bullosa, a skin disorder characterized by fragile skin and blistering. Previous year questions. Which of the following integrins are primarily involved in cell adhesion to fibronectin? A alpha 5 beta 1. B alpha 2 beta 1.